Hi, everyone. Welcome to the session. We'll just give it a minute or two for folks to log in and get set up. Okay, I think for the sake of time, let's get started. So welcome everyone to our inaugural Summit Speaker Series. I'm very glad to welcome you all. My name's Megan Mahoney, and I'm the Director of Scientific Affairs and Training Programs at BioCanRx. And today's session will be called Building a Trans-Canada Highway for CAR T-cell Therapies, and we'll be featuring quite a few speakers that were involved in the initial efforts to get this underway. To begin today, we'll be having a session that will focus on the patient perspective, and will also provide a plain language summary of CAR T-cell therapies. This is ultimately oriented toward patients and caregivers, advocates, and anyone else who's interested in learning a little bit more about the potential of CAR T-cell therapies. So in terms of a few technical notes, as you can probably see already, during the presentation, you will be muted and have no video. But if you have questions, please do feel free to ask them as we're going along in the Q&A box at the bottom of your screen. You'll also be able to answer or type your own questions and upvote other people's questions using the thumbs up functionality. If you experience any sort of technical difficulties or have any sort of technical related questions, please use the chat function at the bottom of your screen. So at this point, I'm very glad to hand it over to Julian. Julian, I'll stop sharing my slides and then you can share yours.
Perfect. Does that look good, Megan?
That looks great. Yeah, perfect.
Awesome. All right. Well, good morning, everyone. Thanks for joining this presentation. And it's here on the West Coast. So I've been asked to give a kind of a brief introduction to CAR T-cell therapy and the program in general. So I like to always start these kind of talks with these images where I think really highlight how dynamic and interactive these therapies we're building actually are. So in the white here is a CAR T-cell, and the pink cells here, down here are these tumor cells. You can see these kind of molecular structures that are actually going after and targeting these cells. And this is kind of counterintuitive therapy.
Hi, Julian, I'm sorry to interrupt you right off the offset. There's quite a bit of noise coming from your end. Could that be an audio issue, a headset issue? I know we had that during testing.
Yeah, it's coming from mine. Let me drop my mic. Maybe that's gonna. Can you still hear me fine?
That's much better, thank you.
Okay, perfect. Get rid of this. All right, back to, yeah, so as I was saying, these are the kind of therapies we're designing. They're really different than the typical kind of drug structures you might think of as chemicals and molecules, but really they're living organisms that we're building here. Next slide. All right. But really to give a kind of brief introduction and what these actually are, because you probably hear this word CAR be bounced around a lot and what this actually means. So what this is a chimeric antigen receptor. And really what we're doing on the fundamentals is we're taking components of immune cells, specifically these T cells, and things called antibodies. And these are very specific proteins that just like to bind to other proteins essentially. And they have these little domains here that are just really sticky and highly specific for what they're targeting. So this has probably been a lot in the popular media right now with COVID and designing antibody therapies. But in CAR T-cell therapy, what we're doing is we're picking parts that are good. So the signal 1 and signal 2 from these proteins and T cells, and these antibodies, identifying the parts we want. We make a blueprint and we package that blueprint. And all that really means is we write it in a DNA or genetic code. That's how we design all these things in biology. And we package that into a virus. And then together this delivers this blueprint to the T cell. So here the virus gives that blueprint to a T cell. And then the T cell's job now is to assemble these parts that we told them in a specific order that we put it in that blueprint. And then at the end of the day, what you have is a T cell but now it expresses this chimeric structure of this variable domain that we took from an antibody and these two signaling domains we took from a T cell. And what this does essentially is allows the T cell to see a specific target really well, and then it gives it this activation signal. So it gets to turn them on essentially. All right, I missed one. So what this actually looks like in clinical practice and how we produce these on a large scale is starting with a patient, we would collect a leukapheresis product from that patient, essentially just a really giant blood draw, isolating the white blood cells from that. Those are the good immune cells, those T cells. And then we give that DNA blueprint in a virus, and we use a system called the Clinimax Prodigy. And this is an isolated system that essentially takes going from 1 million cells to hundreds of millions to billions of cells at the end of the product. And what you have then is an IV bag, just expanded, tons of these cells, these CAR T cells. And this IV bag can then be fed back in through an IV into the patient, and then that therapy is applied that way. So how do CAR T cells actually do their job once they're into the patient? So like I was saying, when we inject these CAR T cells, they actually now have to find their way throughout that patient's body and immune system and identify and locate that tumor. And then once they do, and they reach that area of where that tumor is, they undergo a very specific reaction. So these T cells, via that CAR receptor we put into them, now recognize a specific target. So in the context of what's commonly been highlighted with CAR T cell therapies is B cell malignancies, and CD19 is probably the common target that people would recognize, or CD19 CAR T cell therapy. So when, in this context, when that T cell recognizes that target, it now gets activated, releases all these molecules that essentially kill this target cell on site. And then this CAR T cell can go to other tumor cells, start killing those, etc. And you get this really strong immune response against these tumors. So that's all well and good and how you generally generate CAR T cells. In Canada, we like to do things, a certain way. And how do we make our own therapies? Well, if I can get my mouse to work. Well, we add maple syrup to them. Essentially, that's a secret ingredient to all Canadian recipes and therapies. If we just pour some maple syrup and there you go, that's Canadian immunotherapy in a nutshell. But actually what we have is really, in true Canadian fashion, we generated a very collaborative network. So working with groups out in Ottawa on the east and then in Vancouver and Victoria, we have all these different components to actually making these CAR T cells. So in Vancouver and in Ottawa, we have vector construction, and that's just designing that blueprint that we put into the virus. And then in Victoria, we have CAR T cell production. So that's actually generating the CAR T cells themselves. In Ottawa with John Bell's lab, the vector production is actually happening. So this is actually making that virus that contains that blueprint. And then once these T cells are produced, they get either sent back to Ottawa or to Vancouver currently, where they get infused into those patients and where the patients are recruited. So really in the future, the hope is to have more of these CAR T cell productions and construction sites and more infusion sites across Canada so we can reach as many patients as possible with this therapy. But really what's going to come out of this talk today and what you'll see is that, if I can get to my last slide. That really where we're at right now in the therapy is this kind of early stages. So you can think of these CAR T cells right now as they're really good at doing one job. They recognize their tumor target and they can kill them on site. So like a rotary phone, it can call in, receive calls. It's really not that dynamic at these stages. But where we're moving to in the future with engineering biology and engineering immune cells is really this more interactive software almost driven approach that a CAR T cell can be essentially the possibilities are endless, where you can interact it and design it to interact with its whole environment. It can sense what's around it. It can receive different stimuli. And this is really where the future is going, where we're designing not only new targets, but new ways to modify these cells and make them better at killing these tumor cells. So this is going to come out in the talks today that you'll see the new approaches we're designing here and where the therapy and field is going in general. So I'll actually pass it off here. We won't have any questions or comments yet. But really just to think about when we talk about all these therapies, at the end of the day, this is what we're designing is these living essential cells that we're putting back into the patient's body. So I'll pass it off to Terry now.
Okay. Yeah, first slide. Thanks a lot, Julian. That was fantastic. Those images are absolutely incredible. And I can't even get a family picture right, you know, so just amazing. But thank you to BioCanRx and to Canada's growing immunotherapy network for this opportunity to speak. My name is Terry Harish, and I'm a blood cancer patient living with a rare and aggressive form of non-Hodgkin's lymphoma. I was diagnosed and began treatment about 10 years ago, resulting in a durable remission, but not without challenging side effects that I still manage today. Caveat, however, I have not experienced CAR T therapy. Not many of us have, but I do know what a superior patient outcome should be, and I do know about the potential of CAR T. I should also mention that I've also had the wonderful opportunity to participate as a patient partner, contributing to the design of the CLIC-1901 clinical trial protocol and elements of the phase 1 rollout. So for patients, lay summaries or plain language summaries of topical research is definitely a useful method of translating complex information in a way that's understandable and usable. So today I'll also attempt to provide a very brief, high-level lay summary of the upcoming talks, and then provide some comments and perspectives and questions concerning CAR T therapy. Next. So we'll verbalize these lay summaries. So the first one, Dr. Kekre, she'll present the Canadian-led immunotherapy in cancer clinical trial, the CLIC-1901 we talked about. The first Canadian-based CAR T clinical trial, and is for the treatment of patients with relapsed and refractory blood cancers, specifically harnessing the body's immune system and modifying T cells to see and attack cancerous B cells that express the CD19 antigen. This treatment offers hopes to patients who do not respond to standard treatments and may have run out of treatment options. And complex processes and techniques associated with the manufacturing and the delivery of this therapy will be tested and verified. Dr. Nelson, his study, more timely access to therapy will benefit those blood cancer patients by not having to wait longer for their personalized T cells during a period of rapid disease progression. Building off the CLIC-1901 clinical trial, Dr. Nelson presented an overview of a high-value CAR T manufacturing process, leveraging efficient technology and techniques. And this will enable more timely, less costly deployment CAR T therapy across Canada through point-of-care delivery. Dr. Kevin Hay, so via an enhanced pan-Canadian delivery pipeline, more CAR T therapeutic options can potentially be offered to a greater number of blood cancer patients. Dr. Hay's study looks at building another CAR T design based on a singular CD22 CAR T CD22 construct and a dual-enabled CD19 and CD22 construct for those expressing CD19 negative in their cancerous B cells. Last but not least, Dr. McComb, CAR T therapies that can target many variations of B cell cancer are an important factor in positioning CAR T as an effective treatment option. In this study, Dr. McComb and his team will evaluate several strategies combining CD19, CD22, and CD20 CAR T to identify a therapy that holds the greatest potential for fighting leukemic cells, and that is most clinically feasible in its delivery. Dr. McComb's team also developed a corresponding data set representing its safety and effectiveness, and he will also address the development pipeline focused on CAR T as a therapy for solid tumors. Next. Next slide, please. All right, so what do patients want in a new and improved blood cancer therapy that's emerging from a pan-Canadian pipeline? And I've listed 6 of them, and I also asked the question about how close CAR T is. The 6 outcomes on our wish list as patients: superior efficacy, personalized treatment, minimal side effects, easy access to treatment, minimal out-of-pocket cost to the patient, and that goal that we all have is a return to normalcy and productivity as quickly as possible. And all I can ask and advocate for is that all stakeholders strive to incorporate these common goals in all that they do. And personally, I think immunotherapies such as CAR T are well on the way of achieving that. Maybe not quite there yet, but pretty darn close. Next slide. So we know that CAR T has had some spectacular results so far, and but cutting through some of the foggy mainstream and social media hype can be difficult, and also trying to decipher academic conference proceedings and papers and what's even said during your clinical visits can be difficult too to comprehend. But regardless, I think here are some patient questions that can help cut through some of the hype and the fog, and are probably worthy of hearing some answers from some subject matter experts. So, is CAR T an effective choice for me versus the myriad of other therapies out there right now, the checkpoint inhibitors, antibodies, combo therapies, et cetera, et cetera? What about toxic effects during the treatment? Are they still a serious issue to deal with? It's a new therapy. Long-term side effects, yes or no? Cost, is that going to be a limiting factor? I mean, it'd take us a couple of days just to unpack that statement. And the impact of COVID-19 on the pipeline. It's really curious as to whether or not it's derailed the train on the train track, and hopefully not, but be interested to hear perspectives on that. And how realistic is I guess the blood cancer patient's holy grail, which is, using liquid biopsies and biomarkers to come up with targeted, personalized treatments based on your genetic profile, and maybe delivered via a point-of-care delivery system to a clinic near you. Next. So just my last slide here. I just felt compelled to make some comments about knowledge transfer, and just make some points about informed understanding of therapeutic choices. It's really a critical issue to patients, and all the research that's out there. It really shouldn't be held into the realm of the few, but it needs to be transparent. It needs to be understandable, and needs to be usable for all stakeholders for society to benefit. We patients need jargon-free, plain language information, and the key is that it captures therapeutic concepts that will definitely help patients with informed choices. And I know BioCanRx has done some good work with the Learning Institute and Cancer Stakeholder Alliance, and even speaker series like this, and enabling this to help, or enabling this to happen. So yeah, in summary, we need translators. And also, I have to throw in this last comment about patient engagement in research being very, very important. It enables two-way knowledge transfer versus knowledge transfer in one direction, and leads to better patient outcomes. So thank you very much for listening, and I guess over to you, Megan.
Indeed. Thank you, Terry, and thank you, Julian, for those presentations. And also, both of you are staying at 3 o'clock. So from 3 o'clock to 3:15, we'll be coming back to plain language descriptions of both what you've spoken of so far and also of the speaker talks that will be happening next. So I'll just share my screen very quickly once more, just to go over a few housekeeping notes. So as a start, I'd like to highlight session 2 that will be taking place on December 9th, 2020 at 1:45 p.m. EST. And this will actually be, in some ways, a continuation of what you'll be hearing today. So on that session, it'll be more about the clinical, social, and economic impacts and implications that bringing CAR T therapies to Canada will have. So we'll be talking a little bit about the role of engaging patients, and Terry will be joining us again for that session, as well as other considerations that are taking place as this kind of therapy becomes more accessible to patients. I'd also just like to highlight the agenda for today's session. So we are on to the talks by Natasha Kekre, Brad Nelson, Kevin Hay, and Scott McComb. And again, there will be a Q&A both by those speakers and again by Terry and Julian. And just on a technical note, I think we've covered this, but for those of you who are joining us at 2 o'clock, just a reminder that there is a Q&A box at the bottom of the screen where you can submit your questions and have others upvote. So there are 2 questions so far. Thank you very much. Please do keep adding to them, and we'll answer those at the end of the 4 talks. And so without further ado, I'd like to pass the mic over, so to speak, to Dr. John Bell, who will be welcoming our next speakers. And so, John, I think you're on mute. If you want to unmute and perhaps share your video. There you are. Hello.
Okay. There we go. That's good. Okay, great. Thanks, Megan. Yeah, I'm John Bell. I'm the scientific director of BioCanRx, and first of all, just thanks to all of you who've logged in today to watch this inaugural speaker series that we're holding now in the middle of this pandemic. So it's really unfortunate that we've had to go virtual, but many groups have had to. We're looking forward to next year. Next year is going to be great. We are going to have a meeting in Montreal, we believe, especially if the latest results from Pfizer, Moderna, and I think next week AstraZeneca hold up. I think we're going to see a vaccine in effect. We'll all be able to get together next fall in Montreal, and that would be awesome. Great speakers, so it's going to be a good meeting. But it's great that we have this meeting anyhow, where we can at least connect with each other, share what our investigators have been working on over the last year, and continue on. It's actually an interesting juxtaposition, because what you're going to hear about today is a program that started at BioCanRx as a result of our live meeting that we had in Halifax. And so it just sort of speaks to the fact that these live meetings, these are great meetings, the online ones, but the live meetings are so important because that's where a lot of things happen. A lot of interactions get spawned, and I think you can hear from Natasha exactly how we really started this off several years ago now. But today it's going to be all about Made in Canada initiative. We're going to talk about how it started and how we're going to move forward. And it's gone from a paper napkin to some really nice collaborations across the country with multiple cancer institutes, cancer centers, and the NRCs involved now too. So it's been really a great outcome for all of us. So now I'm just going to hand it over to Natasha, who's our first speaker, and she's going to kick us off. Natasha.
Great, thanks, John. I'll just share my screen here. All right. All right, so thanks everyone for joining us. I'll try to keep this very brief. It's really just meant to be an introduction to the other talks that are coming. And I see from the list that there's lots of familiar names, so hopefully some of this people will know. So just to kick off what John was talking about, we had met many years ago now in Halifax over a beer and realized that, CAR T-cells, which I'll get into in a second, looked like a great promising new therapy, and that in Canada we had the right people to bring together, through the BioCanRx network to hopefully build our own Canadian-made CAR T-cell program, and that's basically what we've done now. So CLIC stands for Canadian-led Immunotherapies in Cancer, and CLIC-01 is the name of our first trial, which is now currently running a trial with a CD19 CAR that we call CLIC-1901, that's the name of the product, for patients with relapsed and refractory CD19 positive hematologic malignancies. And so I won't really belabor this because I think most people were on the call earlier with when Julian was speaking to this. So basically CAR T-cells allows us to arm the immune system of a patient against their own tumor cells. And we do that in the lab by growing their T-cells with this chimeric antigen receptor that we code for into their cells and allows for them to specifically target their cancer cells. So the need for this is huge. I'm a hematologist. I treat patients with blood cancers, such as acute lymphoblastic leukemia and lymphomas. And some of these patients, when they don't respond to standard therapies, have a very poor outlook, and their survival can be less than 6 months, as you see in this curve on the left. And that is obviously a really awful position for us to be in with our patients. And so something like CAR T-cells came along, and we were seeing 6-month survivals as high as 80% in certain trials. And that was incredibly promising very early on. And that's where the excitement around CAR T-cells came from. And in lymphoma, such as diffuse large B-cell lymphoma, again, we've seen remissions in patients up to over 50% in patients who really had no chances of outcomes such as that with standard therapies. These are patients who were failing standard lines of care. So it took a long time for CAR T-cells to come to be, 10+ years of preclinical work and concepts and designs. This is a slide I stole from Dr. Holt, who really founded the beginning of our manufacturing program. And really we decided once that stuff had already been developed in the preclinical world that we could maybe learn from that and move forward with the regulatory side of trying to build an academic-led CAR T-cell program. And that's basically what we did. So the second half of this slide shows the work that went into generating manufacturing that's clinical grade for an actual clinical trial. These are the things, the steps needed to get Health Canada to approve your clinical trial for a completely Canadian-made CAR T-cell program. So this was our trial that I spoke about actually last year at this meeting. We spoke about autologous, this Click 1901 product for patients, adult patients at the time, for acute lymphoblastic leukemia and lymphomas. To be eligible, you had to have failed standard therapies. The first stage of our trial was really to look at safety and feasibility. And the second stage of our trial, once we have enrolled enough patients and shown Health Canada that this product is safe, would then be to look at more efficacy outcomes. And because we get asked this a lot, just to mention that this is not a frozen product, we did this using a fresh product. And we had a dose adjustment in place for patients with really high disease burden. We would give less CAR T-cells. And that's based on previous literature that maybe that would be useful. I won't belabor this point. We've established lots of resources. I've talked about that at other BioCanRx meetings, that we really had to build a lot of infrastructure to get to where we are today. But I think the part that people want to hear about is where we are now. So our clinical trial did open in 2019. We have been enrolling patients both in Ottawa and in Vancouver quite well, actually. The pandemic did lead to about 100-day pause in the trial. Unfortunately, our cells have to be shipped across the country and does require a lot of hospital staff and research staff, and all of that sort of came to a halt when the pandemic first started. We did have a lot of physicians advocating to reopen the trial as well as patients, and that did allow us to open up, we think, pretty quickly actually into this pandemic. So we've been open again after about a 3-month pause. In terms of where we are with the clinical trial, as per any clinical trial, there's always amendments, and we have written our first amendment and submitted it to Health Canada, really to just improve upon things for patient care throughout the trial, make things as streamlined as possible. But most notably, we have broadened our eligibility in this amendment to include mantle cell lymphoma and other types of lymphomas, which really came from physicians and patients asking for us to consider that, and we have now. We do have a data safety monitoring board who meets regularly to ensure that our trial is safe and progressing safely, and they've met multiple times now and been happy with the progress of our trial. And in terms of manufacturing, which you'll hear about from Dr. Nelson, but we are hoping to expand manufacturing of the cells to other sites. So specifically just the CAR T-cell manufacturing part. We are hoping to expand that from Victoria to include now a second site in Ottawa, hopefully soon, and we're underway with that as well. So I'm hoping to have results that we can actually show, hopefully early into the new year. As you know, with any clinical trial, you have to meet certain landmarks in terms of how many patients are treated, how long they've been treated before you show results, but we are hoping to have some information to share in the new year. And with that, I'll pass it off. I just wanted to show that we have a huge team behind this program. Like I said, Dr. Rob Holt was the leader of our initial manufacturing stage. There's all the PIs here in bold, but two names that I think don't get enough recognition are Maruna, who really ran the quality program of making sure that everything we did was up to a certain standard that would be required by Health Canada, and Vari, who really was our program manager and made sure that everything ran smoothly to the clinical trial application, and even now as we move forward. And so both of them we owe a debt of gratitude to. So, and a lot of other people. So with that, I will stop talking super quickly and move it on to the next speaker. And of course, we'll answer questions after the talks. So thank you.
Great. Thank you, Natasha. I'll just jump in, I suppose. Hi, everyone. I'm Brad Nelson. I'm based at BC Cancer in Victoria. And I'm going to be talking about the actual manufacturing side of our program. So I'm going to start by asking the question, or addressing the question, why would scientists want to get involved in manufacturing at all? I mean, normally as cancer researchers, we're busy in the lab discovering new things, new pathways, new drug targets, and we leave the manufacturing side to industry. But in the case of CAR T cells and engineered cell therapies in general, there's a real opportunity here to make huge headway, huge advances against some of the toughest problems in cancer. And to get there, we're going to need, we believe, our own manufacturing capacity so that we can get our innovations out of the lab into the clinic in a timely manner. So Terry, a few minutes ago, gave the patient's wish list for CAR T cells. I think it overlaps. It's probably identical, different wording perhaps, with the scientist's wish list, which I've shown here, is that we want lower toxicity. We want to be able to apply this to other types of cancer. We'd like better penetration of solid tumors, fine-tuned control mechanisms, fail-safe stop mechanisms in case problems arise or the CAR T cells aren't needed anymore. And we need to keep this affordable, feasible, and sustainable if our patients are to have access, especially in a publicly funded healthcare system such as our own. So many of us involved in the program have ideas for how to address these challenges, and many of you on the call as well in your lab research have great ideas. And so with CLIC, what we're trying to do is create a pipeline or platform to get these ideas into the clinic. So the basic idea here, as has been highlighted so far, I'll just sort of emphasize some of the other points, is that there's the manufacturing is in a few different components. One is we need DNA plasmids that encode the CARs, and so Rob Holt's team at the Genome Sciences Center of Vancouver has set up clinical grade manufacturing for plasmids. We need lentiviral vectors to introduce CARs into T cells, and so that's being led by John Bell's team at the BMC in Ottawa. And so these are what we consider centralized manufacturing activities. They can just be done at one place in the country, and these products can then be shipped anywhere. For the actual cell manufacturing, because that needs to be done in real time and with time pressures, the model we're setting up is a local point of care manufacturing model, which I'll describe. And we've started this at our research center in Victoria at BC Cancer, but we're doing it in a way that it can be expanded to other centers in Canada or beyond who want to participate in our network. And really the idea here is similar to the way hematopoietic stem cell transplantation is already done with local processing, manufacturing of cell products at local hospitals. We just want to build on that idea. And by doing this, we will be building Canadian expertise and infrastructure, which will be creating jobs in the knowledge economy, as well as I've emphasized, fostering innovation. So how do we manufacture these cells? Well, the manufacturing platform we are using, and I'll describe in a few minutes in more detail, is called the Clinimex Prodigy. And we think this is really very well suited for this point of care manufacturing model. It's largely automated, although there's still a lot of people involved, but the instrument itself is largely automated, and it's a closed system, meaning that sterility is assured. And with this kind of instrument, the protocols we're developing are much more readily transferable to other Canadian centers. And of course, we've started with our CLICO-1 trial using CD19 CAR T-cells as our initial proof of concept for this. So for us at BC Cancer, getting to this point where we're actually making CAR T-cells for clinical use was something that we started back in 2016. It took a number of years to get the infrastructure in place, the people in place, figure out the regulatory side of this, get the funding that's been required along the way. And so this just gives you some idea of the major milestones that needed to be achieved over what was about a 3-year time frame to open the trial first in Ottawa in 2019. Here's a shot of some of our team members in Victoria, and some of the folks from Vancouver had come over for this day, so they're in there as well. And I'd really like to highlight John Webb, who leads our manufacturing team, Marina Bala, who's been mentioned, leading quality, and there's Vari Sigris, the project manager who's held all this together, Rob Holt, and Kevin Hay, other PIs. I'd like to mention everybody here. Everybody's playing critical roles here. And here's a shot inside our clean room. It's the Conconi Family Immunotherapy Lab in Victoria. There's the team in action. And here's a couple of our Prodigy instruments loaded up with patient cells, CAR T cells being manufactured. And this is a close-up of the instrument, and I'll go through a few of the steps, tell you a bit more about it, but this gives you the visual idea of what goes on here with this automated cell manufacturing. So what are the steps to make a CAR T cell? Well, it starts with a blood sample coming in, a leukapheresis product, a large blood draw. We then need to separate out the T cells from the other blood cells. The T cells are then activated. They are transduced with the lentiviral vector. This is how the CAR gene is introduced into the cells. And then we need to expand the cells to a large number, a sufficient number for dosing the patient. And that then becomes the final cell product, which is sent to the clinic for administration to the patient. So the way it works, here's a picture again of the Prodigy instrument, and then this is just a diagram showing the parts and how they're connected with these tubing sets. So for each patient, we have a single-use, closed, sterile, disposable tubing set that gets wired onto this instrument, if you will, and the instrument has a number of valves, magnets, a culture chamber, etc., where the cell processing actually takes place. So things start by hanging the leukapheresis bag with the incoming blood product from the patient is hung at the top of the instrument there. There's a magnet that's built in here, which is used for the T-cell enrichment stage. So that's where the T-cells get separated from the other blood cells. There's this really cool chamber called the centricult. Very ingenious design. It's a little sort of round-shaped, almost like a bowl, which can centrifuge the cells as well as serve as a cell cultivation or tissue culture chamber. So the cells spend a lot of their time here, and various ingredients are brought in at the right time, right place, for these critical steps that I've talked about, activation and transduction and so on, as well as a number of media changes to keep the cells happy, fresh media, fresh cytokines, that sort of thing. Of course, it's all run by computer, and so at the end of each run, or during the run as well, there's an electronic report being generated. So you have an electronic summary of every action that was performed and every consumable that was used. And this is just a example screenshot of the kind of information it is given. So it's very easy then to track the progress of each run. And if there are any glitches along the way or irregularities, one can troubleshoot them quite rapidly. So this is just an example of some of our early runs done back in 2018, 2019 on the Prodigy. These are different starting products from healthy donors initially, and just showing that we were very reproducibly able to, over it's a 12-day manufacturing cycle on the Prodigy, that we're able to hit or achieve our desired yield of CAR T cells at the end. And this is what it looks like from beginning to end in terms of the actual cells. So this is an example, a picture of an incoming leukapheresis product from a healthy donor in this particular case. And this is what a CAR T cell product looks like at the end of our production run. And that's essentially ready to then be shipped to the clinic for administration to the patient. So what are the next steps for us in our manufacturing facility at CFIL? Well, we're continuously doing process improvement. We're optimizing our logistics, or some of the manufacturing details, the QA, QC components, infrastructure training. You know, we're learning with each run and always fine-tuning our methods. We're also setting up to do correlative studies. We're establishing state-of-the-art immune assays so that we'll be able to evaluate our patient samples from CLIC 01 and beyond, and really start to learn what's going on with these CAR T cells once they're back in the patient, and what are the ingredients for successful CAR T therapy. We're also involved in leading the national rollout of this, so transferring our manufacturing and quality expertise from Victoria and Vancouver to Ottawa and other centers, as has been mentioned. We're also now getting ready to manufacture new cell products. We'll hear next about the CD22 CAR for the CLIC 02 trial. Rob Holt's group has a KRAS TCR that we're starting to plan to manufacture, and many others on the way. And we're also incorporating new technologies. So we're getting a third Prodigy in that has an electroporation unit associated with it, which will allow us to do CRISPR gene editing, as well as other new engineering methods. So we continue to be very busy and very excited to be part of this. So I started with a picture of the Model T manufacturing. You know, this is kind of where we're at with today's CD19 CARs. Where we're trying to get, and hopefully in less than 100 years, is this kind of idea of, here's Elon Musk's Tesla being carried into outer space on his private rocket. These are the kind of things that are possible in the cell engineering space. And we really hope that the platform we've put together is going to enable this in Canada. And I'll just end with my acknowledgements. I'm just focusing on the BC cancer side with this particular slide. I think you're getting that this is really a big national effort, but the cell manufacturing, a big shout out to John Webb, who has led this program. I'm really speaking on behalf of him and the entire team. Kevin Hayes, our medical director for facility, and Julie Nelson as well, a leader in our manufacturing. Our current staff, some prior staff. On the quality side, I've mentioned Varuna, Rob, and here are other quality team members, and the administration side as well, which is so important, as well as the funders that have helped us build the facility in Victoria. And I'll stop there.
Okay, so I think we can now move on to my segment of the talk, which is really a lead on of what Natasha has described, and subsequently Brad has described as well, which is CLIC 02. So going from CLIC 01 to CLIC 02, and I had suggested this idea of calling it the Trans-Canada Railway, just thinking about how important the railway has been for the birth and the growth of Canada, and seeing that in a similar way for this project across Canada. But then I thought, well, how do you talk about cars and railway afterwards? And I realized other people's ideas of a highway might be better. But then I thought, well, railway cars have trains. Railway trains have cars to them. So I subtitled this one called Adding Another Car to the CLIC Train. to looking at trying to use CD22, a different target, to develop CAR T cells for the treatment of B-cell cancers. So just by way of disclosures, I have provided advisory board services to various companies here, as well as spoken and received remuneration from Jazz Pharmaceuticals and some research funding from Janssen. However, I will be discussing an investigational product that is not a product produced by any of these companies. So I first start to say that really the only way we could do such a project is really because of all these great people who have been mentioned already numerous times in the last two talks across different areas in Canada. So you do see here that we have people in Victoria, people in Vancouver. With this project, we're also bringing on board the Children's Hospital as a clinical site, because a pediatric population is in need of these CAR T-cell therapies as well. And working also with the Ottawa Hospital Research Institute, as you see, but also the National Research Council of Canada, which has been key to having this proposal move forward and to getting it towards the clinic. So, you saw these curves basically from Dr. Kekre's talk, where you see the patients with relapsed refractory B-cell malignancies really have a dismal prognosis with standard chemotherapies that are used prior to the advent of CAR T-cell therapy, where, by 6 months, unfortunately, more than 50% of patients typically have passed away in this relapsed refractory setting. So very difficult disease to treat, B-cell acute lymphoblastic leukemia and diffuse large B-cell lymphoma once they have relapsed and are refractory. However, CD19 CAR T-cells have demonstrated significant efficacy in these diseases. So you see, for example, here two of the commercial products, one tisagenlecleucel in B-cell ALL, for example, showing significant improvements in event-free and overall survival. And you see for diffuse large B-cell lymphoma, active-captated cytolucel, just as an example here. There are many other products as well, but showing that we've improved the overall survival up to the level of about 50% of patients still being alive out past 2 years or so. However, this does mean also that despite the promise of all these therapies, obviously CD19 CAR T cells in and of themselves are not the be-all, end-all, and final as Terry kind of mentioned near the beginning, kind of final cure for all the cancers in all the cases. In that there's still this unmet need of, you see approximately 50% of patients who either are refractory to CD19 CAR T cells or relapse after CD19 CAR T cells. So any future of these programs that we want to develop needs to take into account these patients who are failing to respond to CAR T cell therapy and trying to find ways to actually be able to target and achieve a cure in some of these patients. So this highlights a need for further therapies or approaches to CAR T cell therapy. Now, one of the challenges in B-cell cancers is that CD19 negative relapse is a common mechanism of escape from CAR T cell therapy. So this is from a large CAR T cell therapy trial that was done at Fred Hutchinson Cancer Research Center. And you can see that there are patients who have CD19 positive relapse and patients who have CD19 negative relapse. It's approximately 2/3 in this trial who had CD19 positive relapse and 1/3 who had CD19 negative relapse. And what you can see here from these graphs is basically that this color here shows that the CAR T cells are still in the body. And so what you're seeing is that the CAR T cells are still hanging out in the body before the point, up until and during the point at the relapse. You see from the little cross here. So you can see that the cancer is coming back while there are still active levels of CAR T cells in the body that are directed against CD19, and so you can get this CD19 negative escape. So where the cancer has found a way to hide CD19 from its surface or not even express CD19 on its surface so that it can evade being killed by the CAR T cells. Often in the case of CD19 positive relapse, it has been that the CAR T cells somehow, so you see from the gray here, this is when the CAR T cells have become undetectable. You can see that the CAR T cells have been lost in the body, and then they relapse. Tends to be more common. But basically, you would need something that would be able to work in potentially both of these scenarios if you're focusing on trying to improve the outcomes in those patients who have relapsed after CD19 CAR T-cell therapy. So any novel CAR T-cell therapy approach in this sort of setting for these diseases needs to address CD19 negative escape. So this comes back to the idea of, well, what makes an ideal target for CAR T-cell therapy? Well, it's fairly simple. Basically, you want it something that's on all cancer cells, if possible, and absence on a non-cancer cell or absence of those normal cells is tolerated and manageable in the body. And so CD22 is actually a very good target in this way, much like CD19, where it is present across, a spectrum of B cell maturation, but not on any other normal cells of the body. But it's also present on, the acute lymphoblastic leukemia, CLL, and various different B cell lymphomas. So this makes CD22 an additional good target for B cell cancers. So what we did is we did a systematic review of CD22 CAR T-cell therapy that's out there currently, as there are some trials running down in the US using CD22 CAR T-cells, either alone by itself or in combination with CD19 targeting. And so we found 14 unique trials that are reported in the literature representing 330 patients, and showed that single targeting with CD22 CAR T cells in B-cell ALL had a complete response rate of 66%. And in a lot of these cases, these are also, some patients who have already seen CD19 CAR T cell therapy. So demonstrating that this therapy can work even in these patients who have relapsed after CD19 CAR T cell therapy. Now, the next question you ask is not just, is it effective, but is it safe? So, two of the known toxicities of CAR T-cell therapy are cytokine release syndrome and what we call immune effector cell associated neurotoxicity syndrome, or ICANS for short, because, doctors like to make acronyms out of everything. So cytokine release syndrome, or CRS, it's this consequence of this immune activation that occurs by putting all these new immune cells into the body and then, basically being activated by the cancer and then activating the rest of the immune cells that are present in the body. And this is typically now treated with tocilizumab, which is an antibody that blocks the receptor for this cytokine called IL-6, plus using corticosteroids. So this is fairly well known in the literature to do this now and used clinically all the time with CAR T-cell therapy. Now, what we saw is that from this systematic review we did is that across different studies, but focusing on those ones that were CD22 targeting only, we saw that, a lot of patients did develop CRS, and this is actually fairly typical and fairly similar to what's seen with CD19 CAR T-cell therapy, where, approximately somewhere between 70 to 90% of patients get CRS. But to get to the severe form that potentially ends you up, in the ICU, needing some blood pressure supporting medications, things like that, really, it's very rare for that to happen, and happening in only about 3% of patients or so, which again is fairly similar to what is seen with CD19 CAR T-cell therapy. Now, when you look at the neurotoxicity, or ICANS, this is just broadly called ICANS because it can present as a various different types of neurologic findings. So some patients, they simply have trouble writing, or they have a tremor. Other people can potentially, have seizures, or in more severe cases, or end up with a coma that they subsequently come out of. So this is typically treated with corticosteroids. And actually, you know, I described to a lot of patients that often sometimes it's more scary for the family to see this happen than it is for the patient themselves, because often they just kind of wake up out of it and then they kind of say, okay, well, what happened there? But, you know, it looks like, you know, neurotoxicity happens only about 14% of the time with CD22 CAR T-cell therapy. And severe events are only about 1%, where they might actually end up with, you know, seizure or potentially a coma or things like that that they would then come out of. So again, you know, it's important to note that severe events can occur, but again, this is similar, if maybe better, than CD19 CAR T-cell therapy, although we're not directly comparing them using this method. But it's a good signal that this idea at least should be relatively safe and similar to CD19 CAR T-cell therapy. So, you know, the next stage, well, we need something to bind CD22 to make a CAR. And so this is where the NRC came in, and, you know, these slides are courtesy of the NRC and the group there who you'll hear from next. But essentially, they have this very nice pipeline now for making monoclonal antibodies and then developing them into CARs. So here they, you know, they immunize actually llamas here to make what they are called VHH antibodies, or single domain antibodies, isolate out those cells, extract the RNA from there, amplify it up, and then screen it against CD22, and are able to come up with these binders for Scott McComb, who's going to talk to you next, to actually try out in various different CAR structures before taking them to us. And so this is what they have done already for our single domain CD22 CAR T-cell trial. And so they had 22 binders, and they've honed it down now to 4 binders that we are currently actively screening as our leads for taking forward into the clinic. So we're kind of right now at this stage here of where we're actively progressing to confirming which of these 4 will be the best binder to take forward into the clinic. And there's a few various different things that you know, need to go into, you know, what makes a good binder, but basically you want it to bind the target and you want it to not bind anything else, so that you want the off target to be negative, you want the on target to be positive. And this tonic low means that if you don't show it to any sort of stimulus, you want it to not be proliferating a lot on its own, basically, and trying to take over, which can sometimes happen if there's some issues with how these are the CARs are designed. So basically, that's what this whole screening process went through here. And I'll show you some of the final last data. So 4 different constructs in these 4 different colors that are being taken forward. And you can see on the top here, when you have a CD22 target cell line versus on the bottom, when you have a CD22 negative cell line, you can see here that those CAR T cells expand, and they're able to rapidly kill the target tumor cells, compared to here, the tumor cells continue to grow if it's just regular T cells and not that don't have a CAR on them. And you can see here, though, that if you don't have CD22 on the surface, the CAR T cells do nothing and the tumor continues to grow, which is what you would completely expect to see if it's not supposed to be targeting anything other than CD22. So what is the rationale then really for making a Canadian trial of CD22 CAR T cells? Well, we have manufacturing and clinical expertise with producing and administering CD19 CAR T cells, or the CLIC 1901 product, as that is running through the CLIC 01 trial that Dr. Kekre explained, and Dr. Brad Nelson explained the whole manufacturing process. I hope I've demonstrated to you that there's a need for therapies after CD19 CAR T cells, and that CD22 appears to be a safe and effective target with no unexpected off-target toxicities in clinical trials so far. So we propose a single-arm phase 1/2 stage trial of CD22 CAR T cells for the treatment of relapsed refractory B-cell cancers. And this is what we've taken forward to Health Canada, and are approaching them to do this as a trial. So the objectives we've laid out for the trial are, the primary one is to determine what we would call the optimum biologic dose. As this is a new product that is being tried for the first time, we'll try a few different doses in our first phase, the first step of the trial, and then hopefully take this forward to a phase 2 trial with the correct dose that we've determined based on this. And then secondarily, we want to confirm that we're able to actually produce and administer these cells using this same point of care manufacturing system that Dr. Nelson explained to you. And then look at how effective are these, and also any toxicities that may result from this, as well as to see what the CAR T cells are doing in the body. So we're including patients with B-cell cancers. So you see here that they have to have B-cell acute lymphoblastic leukemia, chronic lymphocytic leukemia, or a B-cell non-Hodgkin's lymphoma, and have to have relapse or refractory disease, which we've defined here. So this is fairly standard across different CAR T-cell trials. And any participant needs to have documentation of CD22 expression demonstrated on the tumor itself, because as this is a CD22 targeted therapy, we want to make sure that we're administering it to those patients who do have CD22 on the tumor cells. And also that they would be able to tolerate the chemotherapy and the cell infusion and any potential issues with toxicities. And that's what we call that adequate organ function, which is we've predefined. The one thing I wanted to point out here is that we are, as I mentioned, including pediatrics, hopefully in this trial, and looking at 1 year of age or older to be included. And this is based on the fact that, as I mentioned, pediatric patients with B-cell cancers are a patient group that is in need of these therapies, and we really want to make sure that we can be as inclusive as possible. So what's the current status of this trial? Well, we've already held what we call a pre-CTA meeting with Health Canada. So this is where you meet with Health Canada to discuss some questions you may have before you actually present the full, what we call CTA or clinical trial application to them for full review. And so, we had this pre-CTA meeting where we had some questions answered and have a good path forward on the basis of that. And we're looking at choosing that lead binder out of those 4 by the start of the new year here, and then subsequently throughout the new year, actually make some of the virus and do some clinical manufacturing runs and submit the final clinical trial application at the end of that year to Health Canada, and aim for a clinical trial opening in 2022. So that's basically the path forward, and really couldn't do it without all these people who have been working hard on this project. And I realized I had to redo this list from the other ones, and it's a quickly growing list. So if you don't see your name on this, I apologize profusely. I made up this list late last night. So again, I apologize if you're not on this list, but really everybody's important in this large team of ours. Thank you very much.
Hey, Scott, you're muted.
Thank you. Sorry about that. Yeah, just thank you to the organizers. I'll hustle up here as we're cutting close on time. I'm speaking today representing a large team at NRC that's working on CAR therapies, so I'll just acknowledge that. we're very much on board with this idea of trying to build a cross-Canada network, you know, a railroad or you know, highway, whatever you want to call it, something to link cell therapy and provide cell therapy access across the country. We at the Human Health Therapeutics Research Center have expertise in generating antibodies, and we wanted, quite a few years ago, we started getting interested in cell therapies and to see whether we could use our antibody expertise in order to generate new novel CAR therapies. And so we've been trying to build what would be a CAR factory at NRC in order to be able to build trains that could run on this network of cell therapy hubs across the country, and so that we could bring new molecules, new therapies that would be accessible to Canadians. And so this is actually part of an NRC challenge program for disruptive technology solutions for cell and gene therapy. This is a new program that launched last year in order to enhance NRC's ability to collaborate with clinical, industrial, and academic collaborators across the country, and to be able to actually provide some funding to our collaborators in order to help to develop those links and in order to help us build new solutions for cell therapies. And the mandate really is to increase accessibility and affordability specifically. Jumping right into what specifically I've been doing as part of the cancer immunology team at NRC, we've been looking at how we can really focus on that external domain of the CAR. So you had a nice introduction to what is CAR technology already, but the external domain is actually what does the binding, what's able to bind to specific molecule, to cancer targets. And so you can make this out of different things. Typically, it's actually made from an antibody, but antibodies have two chains, a heavy chain and a light chain, and you have to put them both together in order to get an antibody. Interestingly, camels and other species of camelids produce antibodies that only have a single heavy chain, and this makes it actually much easier to engineer these. So that's the reason we have that. In addition to using things like mouse or human antibodies, camelid antibodies offer an easier platform for engineering. And so we've established a platform by which we can easily swap out these antigen binding domains, or the other extracellular domain, which is the hinge, that is the spacer that determines how far away from the cell the CAR is located. And so to swap these out in order to be able to screen them. And I just want to acknowledge that, you know, this is really the front of the train, the locomotive. This is the powerhouse for our cell therapy, but it can go nowhere, you know, on its own. It's really not very effective. It's not much of a train unless you have all of the other partnerships that are essential to make these therapies a reality. So, you know, you really need clinical expertise, patient partners, vector production, all of the elements. And that's where we're really collaborating with great researchers in the BioCanRx network, some of whom you heard from today. So what does screening antibodies really look like? Here's an example of where we took a CD19 CAR. This is a CAR that's very similar to what's already in the clinic in CLICA-1. And we made it so that you could easily swap out the antibody portion of this. And we showed that if you could change that to a Herceptin-based antibody, you would no longer respond to leukemia in red here, but then you would start responding to breast cancer cells. And so you can do this same type of thing to rapidly put prototype CAR molecules, and you can screen them in high throughput using a T-cell line called Jurkat cells. And basically you mix these Jurkat cells with different types of target cells, that may be leukemia, maybe breast cancer, maybe other types of cells, and then you screen for the ones that have the right response properties that you're looking for. And so we actually published an article on this earlier this year, and I encourage people to look that up if they're interested. So then downstream of this, we can discover a molecule that works, has signaling properties and responds specifically to the target. From there, we can select leads based on diversity as far as the sequence specificity or the strength of the signaling. And then we package those into viruses. We produce these in high concentration, and then we can put them onto primary human T cells and then mix them with fluorescently labeled target cells. So similar to the Jurkat assay, but in this case, we're using primary cells. And in this case, then we're looking at, are the cells able to actually kill tumor? And we can also do this in using live microscopy. So we can do this in not as high throughput as we can do with the cell line, but we can do it with high content. So that is to say that we can get a lot of information about how the molecules, cells are behaving. And so we can see different types of CARs. Some might respond more weakly, and some might respond very strongly, but maybe with less specificity. So we can balance those different properties of CAR cells and study their biology, and then eventually come up with optimized therapies. And so we've taken a broad approach at NRC, looking at many different targets. As I said, we're quite capable as far as generating new antibodies. So we've looked at targets specifically for hematological tumors. And this is actually the project that I'm leading in order to develop combinatorial CARs that will target multiple antigens. So for instance, you might take a CD22 CAR and a CD22 or CD20 CAR, and link those in order to create what's called a tandem CAR, where it will bind both molecules and either one will activate the cell. And so through this, we could potentially target leukemia more effectively. And even if they lose something, as Kevin discussed earlier, if they lose CD19, they could still respond against CD20. We're also interested in solid tumor, and I've been working a bit on this with other researchers in NRC, and I'll talk a little bit about this later today. So just, I want to stress that understanding the biology of CARs is still in its infancy. By better understanding CARs, I think we can build much better CARs. And so that's something we're trying to do in sort of a design, build, test, and then understand, and go through that cycle as many times as we can before we get to the clinic. And so here's an example where we've mapped different single domain antibodies that bind to different parts of a CD22 molecule, which is actually quite a large molecule that's on the B cells. And we can then, based on this information, by understanding how they bind, where they bind, we can then use that rationale to build next-gen CAR designs, and we can combine those, maybe double binders. We can make biparatopics, which will bind multiple epitopes on the same antigen, or maybe binding multiple antigens, as I said. And so this is really the impetus for our recently initiated grant funded by BioCanRx, which is to develop multiple develop multi-antigen targeting CARs. And we're going to look into how best to combine these. I discussed already tandem CARs, but what if you had two different CAR molecules? Is that better than a tandem CAR? Is that worse? What if they're on different cells? You know, if you have some cells that are CD19, some that are CD20, is that an advantage? And so we want to really interrogate this in a broad fashion and look at what is the best way to generate these, and then move that along towards clinical application. So now that I've talked a little bit about what we're doing in the hematological CARs, I want to talk about solid tumor CARs. In the paper that I mentioned earlier, we had shown that the development of a mouse monoclonal antibody targeted against EGFR-V3 CAR that can specifically recognize glioblastoma cells. EGFR-V3 is actually a great target for CARs because it's very specific. It's only expressed in cancer cells. But the downside is that it's not as common as things like HER2 or EGFR, which are overexpressed in cancer cells, but not completely specific to tumors. So we were interested in maybe if we could design a CAR that would be more selective for overexpressing antigens like EGFR. So in this case, we used an EGFR-specific camelid single-domain antibody, and we raised CAR T cells that were specific for EGFR. And here I have some videos to show you. In red are ovarian cancer cells, and in green you'll see here are CAR T cells that are going to rapidly recognize and kill the ovarian cancer based on their overexpression of EGFR. In contrast, the CD19 CAR T cell really does nothing against this target because it has no recognition antigen in the ovarian cancer. And so here you see the T cells end up winning. But these can recognize cancer, but the problem would be that they also would recognize EGFR expressed in normal tissue. And we can actually model this with cell lines, a low expressing cell line, EGFR expressing cell line MCF7. We see that our CARs respond to both very well. And so that's a problem. That would create toxicity. And so our approach to try to address this issue was to try focusing on the hinge domain, which I mentioned earlier, which is the spacer for the CAR. And if we shorten this, we found that actually we can reach a threshold where the right size of hinge will actually reduce the response against these EGFR low cells. And here you see with a very long hinge, we see response against both SCOV and MCF7. So any level of EGFR is adequate to activate these cells. However, if we shorten the hinge, they maintain their response against the tumor, but no longer recognize EGFR low target cells. And so we think that these would be much safer. And actually, we found that using single amino acid truncations of the hinge domain, we can actually have precise control over how much signaling we want in a CAR. And so you can really specifically program cells to recognize tumor. And we found that this also applies in a mouse model of ovarian cancer, where we saw that shortening the hinge also decreases response relative to the length of the hinge. And so this really gives us an exact control over how much signal we want. And so we can find what's the right balance for having an effective CAR, but also a safe CAR. And so we've recently released a preprint on this work. So I encourage people to look that up. As I said, we have a broad approach. We're looking at many different antigens. And I think I'm very excited about the future, but having all of these different potential CARs coming down the pipeline, it's going to be likely too many therapies to really serve the demand. And so I think the long-term future, which I'm just going to mention very quickly, is to reprogram cells more completely using things like genome editing, CRISPR technologies, and stem cells in order to actually grow up unlimited numbers of cells that are specifically programmed that then we can just roll out programmed cells. This vision is going to take a while to come to fruition, but I think it's time to start developing these, and I think we'll see that as the future for CAR therapy. So I'll just acknowledge Rossini Viratna is another researcher at NRC who's really led this work with me, and I'll just leave it at that as we're running out of time. So thank you very much.
Thanks, Scott, and thanks everyone else. So we are a little bit past 3 o'clock. I am hoping that we can take a few extra minutes to answer some of the questions that came in via the Q&A. So Natasha's going to moderate those questions, but just a reminder to all participants that this session was recorded, and for those of you who do have to jump off, it will be available on our BioCanRx website and on our YouTube page. So I just want to again thank all the participants for attending and for our speakers for participating. So Natasha, I'll hand it over to you for 5 minutes, and then we'll do we'll keep the plain language summary of this speaker series as well with Terry and with Julian after as well. Thanks, everyone.
Great, thank you. There's lots of great questions. I'll get through at least a few that I think are really great to discuss. So first, I'm going to throw it to Dr. Nelson. And the question for you, Dr. Nelson, is do we have the capacity to produce and manufacture these CAR T cells without industry involvement? And would there be value in having industry partnerships to provide access to expanded technologies, or do you think industry is lagging behind?
Oh, great questions. So I'd really like to emphasize that we do intend to interact with industry, even though we've emphasized this as an academic, publicly funded program. We're setting it up in a way that we do interact with industry. I'll also point out, of course, I talked about the Miltenyi Prodigy system that we're using for our point-of-care manufacturing as one example. So Miltenyi is a company, there's a clear interaction with industry. It's just a different type of industry than what I think is intended by the question, which is more big pharma and biotech. So our hope would be that biotechs with ideas for a next generation CAR of some sort would start to see us as a possible conduit for getting their new technology into clinical trials in a quick and cost-effective manner. So we're absolutely open to those ideas. As far as big pharma, Novartis, Gilead, for example, producing commercial CD19 CARs, we don't see any conflict there. We think it's going to be an ecosystem with those sort of products coexisting with the products we're working on. And really, it's all about making sure our patients have as many options as possible.
Great. And next I'll go to Dr. Hay. A few questions came in specific about CD22. A couple things. Do you think that there are CD22 specific side effects in knowing that there's a low level of expression of CD22 on different cell types? And also, do you think there's value in a strategy where we give CD19 then CD22 versus combining them and giving them at the same time? What are your thoughts on those two questions?
So those are both very good questions. To answer the first one, CD22 expression outside of normal B cells is I would hesitate to say not seen at all, but at least the literature would support that so far. I don't think it's really present as a antigen or a target on any other cells other than B cells, and it's very similar kind of profile as CD19 CAR T-cell therapy, and so I would say it's probably below the level at which there's any concern of it targeting anything other than B cells. Now, B-cell aplasia is something that's known to occur with CD19 CAR T-cell therapy, and some patients do require intravenous immunoglobulins or intravenous antibodies that are pooled from healthy donors to help support them after CAR T-cell therapy, but that seems to work quite well to help prevent patients from getting infections after CD19 CAR T-cell therapy, and I would expect the same sort of potential side effects of what we call off-tumor side effects with the CD22 products. And I think that's held up by those different clinical trials that I mentioned already that show that it doesn't seem to have any other safety signal that's of concern compared to CD19 CAR T-cell therapy. In terms of the subsequent question of would it be good to combine the two or to do them sequentially? Yes, that's exactly what we're thinking of doing. But the initial part of this is to, first of all, just demonstrate the safety of our particular binder that, that is going to be our lead candidate going forward. And once we've demonstrated that that one is safe and we can figure out what dose is safe to use with that, then we can actually design subsequent trials of how we actually combined it with CD19 CAR T-cell therapy or potentially other future therapies as well.
Great. I'm going to quickly go with Dr. McComb for two questions as well. If I can ask, so a lot of people are asking questions related to cost and shipment and feasibility of our system, how it would hold up if we end up being able to do this outside of clinical trial. Some of that we will get into at the next session, so stay tuned for some of those questions. We won't get to that today. I think today we'll focus on the science and the manufacturing. So the questions I would like to ask and answer in the last few minutes here, a question about where exactly is CD22 found. You heard Dr. Hay mention this, but are they found on mural cells and brain vessels such as CD19? And another question was, would there be value in incorporating reporter gene imaging technologies for tracking the cells in vivo? So if you could answer those.
Yeah, so the first question on CD22, I think it's the finding that CD19 is expressed in other cells was rather surprising, and I think CD22 we could have similar surprises. But so far the safety signal has been strong for CD22 targeted trials, so I think we're hopeful that that will continue. But I can't say conclusively that there's no other cells. I think it seems that the toxicity, the off-target risk, is not going to be worse than CD19 target therapies. I don't think CD22 is expressed on mural cells like CD19. On the gene imaging, that is interesting. The difficulty, I think, with imaging, we do that in mouse models for sure. The difficulty is that these gene imaging technologies are often immunogenic. They'll involve use of exogenous proteins that you couldn't really use therapeutically because you would get an immune response against those exogenous proteins. You generally want to minimize how much extra DNA you're putting into these therapies. So I think it would be interesting, and perhaps there's other technologies that in the future could be used to identify where the CAR T cells are going and whether they're getting into tumors in patients. But I think that probably won't be through exogenous genes per se, but maybe other labeling technologies. So maybe I'll leave it at that. I do see that there's one question about humanizing the single domain antibodies in the chat. That is a good question. We're hopeful, actually, surprisingly, camelid antibodies are more homologous to human than mouse, even though they're only a single chain. There's higher homology with humans. So we're hopeful that the humanization won't be necessary. And there are actually ongoing clinical trials with camelid targeted CAR T, and it hasn't so far been an issue, anti-camelid antibodies, but that's something we will be looking for in our trial. And there is potential to humanize them in the future, but we're hopeful that that won't be necessary, and we'll get a strong therapeutic response without that. So.
Okay, so we'll leave it at that. There are great other questions that we just didn't get to yet, and I will hand it off to Terry and Julian, I believe.
Great, thanks again, Natasha, and to everyone else. So we'll take a few minutes at this point. Perhaps, Julian, if you can provide a high-level summary of the talks, any key messages that you think are important from a plain summary point of view, and perhaps as a trainee that you see as important. And Terry, I'd ask you the same thing, so maybe you can make any concluding remarks from your end. And to folks, participants who are still attending, please do write any questions that you have for Terry or for Julian in the Q&A box.
Yeah. So yeah, I would like to thank all the speakers again. I think that was fantastic today. Really, it covered such a wide gambit. I think seeing from Natasha's talk early on and the click trial and CD19 CARs, like, I think one of the key highlights for people to take home too is just how fast this was all done. This was really, like John Bell mentioned, an idea in Halifax not that long ago. And then going from that idea to actually now treating patients is really quite remarkable. And if you think of any drug in a pharmaceutical company or anything, there's often, a decade of lag time between getting a drug from an academic setting into an actual patient. So I think that's really deserves some highlight and recognition, just how fast this collaborative network is moving and how well it's actually working to get to this point so fast. And that CLIC-19 trial has really been progressing really fast. So it'll be exciting to see in the future the results that come out of that. And then Brad's talk with manufacturing, that again, I work in that lab where the manufacturing is going on, and I can speak personally just how quickly that evolved to go from basically an empty lab space into a full-fledged clinical manufacturing space is remarkable. And the team here and the manufacturing team led by John Webb and what they're able to do and produce these products is really remarkable. And then the future of, like, essentially within 2 years, more or less, we built a clinical grade lab space here, and just how potentially easy it is, relatively speaking, to transplant that into other centers across Canada. So really, this is a streamlined approach. It's not something that's going to require billions and billions of dollars and full buildings being redone and all that. It's really something that can be kind of just pick your center and you could probably fit one in there, more or less. And then really going to Kevin's and Scott's talk and the CD22 angle of it. If we just started the CD19 trial not that long ago, but already to be talking about and picking these next targets and getting ramped up to treat patients really rapidly is, again, a remarkable feat, in my opinion, especially just seeing from a trainee perspective and knowing how long these therapies often take to go from a kind of scientific standpoint into actually treating people. So I think everyone should kind of really take that as a hopeful message that with the help from BioCanRx and this network in general, and this group of people, we've been able to take ideas and really turn them into therapies and treating patients and affecting lives quite rapidly. So I think that's been remarkable.
Thanks, Terry. If I could turn it to you now.
Yeah, sure. Everything that Julian said. But I have to congratulate the speakers on their presentations. They're really informative, and even from a lay person listening, like myself, listening to them, I I certainly got a lot of the out of the presentations in terms of furthering my my understanding of how CAR T works and the evolution of CAR T as we proceed down the pipeline. To some of these things that Scott was talking about that are a real laudable goal to follow. I have to say, I've been involved with CLIC-1901 as a patient partner, and I am really happy to see how the clinical trial has evolved, and really what it's done in terms of establishing the expertise, CAR T expertise from the point of view of clinical delivery to manufacturing within Canada and across Canada. I think that really bodes well for the patient community, obviously, and the future delivery of CAR T treatments to handle blood cancers, and notwithstanding solid tumors as well. So, with regards to Dr. Nelson's presentation concerning point of care delivery and the equipment that is being used and utilized and optimized, that will again bode well in terms of helping the delivery of CAR T products to patients in need on a more decentralized basis. Certainly which ultimately means quicker access, high quality product, and a happier patient, or greater number of patients that are happier. And with regards to Dr. Hayes' presentation, definitely CD19 is CAR T is a singular approach to providing therapies, expanding that into to different constructs like the CD22 gives patients more choices as far as therapies go. And again, a very laudable achievement. And I now understand a little bit more about what CD19 negative means. And so thank you for that. In Dr. McComb's presentation, it's completely mind-boggling. Camelids, who knew? In terms of being able to improve and help facilitate the construction of CD19 and CD20 and CD22. So, again, that's a very important factor in positioning CAR T as effective treatment option with these various variations. So, in summary, thank you very much for enabling me to participate in the speaker series. I learned a lot, and hopefully help translate some of the more complex concepts into things that are understandable. Thanks again.
Great, and thanks to you both and to our other speakers, and thanks again for the participation of our audience. I think this is a good opportunity to close, and with the final reminder being that this will be posted on our website, and there will be a survey that you'll receive after you close out of this window, and we really do appreciate your feedback in that survey. So again, thanks everyone, and see you next time. Take care, bye.
Thanks, Megan.